This podcast is about the first part of Recombination, which is linked genes. This is going to be from chapter 10 of your textbook. We'll talk about what recombination is eventually here, but I want to start with the concept of linked genes. So please write this down as your heading. So far we've been talking about genes as kind of like cards in a playing deck. They can be independently shuffled and rearranged to be put into different gametes through meiosis. But in reality, every chromosome actually has more than one gene on it. And so that shuffling doesn't always work as well as what we've been discussing. So genes that are close together on a chromosome, so physically located near each other on a chromosome, are considered linked genes. And the reason we call them linked is because when gametes are formed through meiosis, they usually travel together. In other words, if your eye color gene is really close to your height gene on the same chromosome, those two genes, the versions of those genes, will be put into a gamete together. Okay, they won't be separated like shuffling a deck of cards. Instead, those two cards are stuck together. So if you're like me and you have kids at home that play with your deck of cards and they get them all slimy and sticky, okay, those two cards don't separate from each other when you shuffle them up and put them into different gametes. Instead, they stick together. Okay, in this way, these linked genes are on the same chromosome, and so these genes travel together into the same gamete. And of course, again, this is all during meiosis. So in this way, these genes do not obey the law of independent assortment. Remember back we were talking about Mendel's laws that he established. So the law of independent assortment, the alleles were going to separately divide themselves into different gametes. Well, some of these alleles are stuck on the same chromosome and they're very close together, so they do not divide themselves into different gametes. Instead, they travel together. Okay, so we're going to take a look at what this means for meiosis. First, we're going to look at unlinked genes. This is what we've been doing all along when you've been modeling meiosis. We're assuming that genes A, let's get rid of this quick. Okay, so gene A here has two different alleles, big A and little a, and gene B here has two different alleles, big B and little b. All right, the actual chromosomes are represented by these bars that I have drawn here. Okay, but since they have the same genes on them, these are a homologous pair. So maybe the big A allele came from the mother, and the little a allele came from the father, and the little b allele came from the mother, and the big B allele came from the father. Okay? In each case, or in each example, this is one set of homologous chromosomes. Okay, so maybe we'll say like this set is homologous chromosomes for number 1. So chromosome 1, you have one from your mom, one from your dad. And maybe the other one is chromosome 2, a homologous pair, one from your mom, one from your dad. Okay, so this organism has 2 pairs of homologous chromosomes. Remember, we have 23 pairs of homologous chromosomes, so this gets even more complicated the more chromosomes you add. But we're going to deal with one with 2 homologous pairs of chromosomes. They each have a different gene on them, the B gene or the A gene, so those genes are not on the same chromosomes. Hopefully that makes sense. So when we're looking at meiosis of this individual cell, remember that they're going to double. So let me just kind of walk you through meiosis with this again. I'm going to do highlighted each of these in a different color so that we can track them more easily. You may want to diagram this in your notebook as well, by the way. If you're still struggling with meiosis and how it works, this would be a great diagram to put in your notebook. If nothing, what I'm going to draw here, but if nothing else, you should at least have this diagram that's already set up for you into your notebook. Okay, so this with the 4 chromosomes and then dividing into the 4 gametes. I'm going to step you through, though, how these 4 gametes are made. All right, so let's go through the process. Remember that each one of these chromosomes forms sister chromatids, so I'm going to make sister chromatids for our big A and sister chromatids for our little a, and sister chromatids for the big B, and sister chromatids for the little b. Okay, and they line up in meiosis, remember, across from each other. So I've got them lined up against their homologous pair, right? The A with the A and the B with the B. Okay, they divide in half. So now we've got this set of chromosomes are together. So we've got one A version up here and one B version down here. And we've got this set of chromosomes here and here. All right, then these divide again. So let's do our two separations. Okay, remember this one and this one. We had two cells at this point. So we'll show the result before we divide them again. And that. Okay, so there was our first division after we split that cell in two. Again, they divide a second time. So this time the sister chromatids split from each other. Okay, this one becomes two, and this one becomes two cells. And then we look at what's in each one of those cells. Go back to my colors. Okay, each one of these got one blue chromosome. Remember the blue chromosome represented the big B, okay, up here from the beginning. And then each one of these cells gets a big A chromosome, and that was from up here. Okay, on the other side, each one of those cells gets a little b chromosome, again from where we started, and each one of those cells gets a little A chromosome, again from where we started. So the resulting combinations of gametes, we've got a big A and a big B, and another big A and another big B, and we've got a little a with a little b, and another little a with another little b. Okay, so that takes care of this possible gamete, right? Little a, or sorry, big A, big B, big A, big B, and this gamete right here. Little a, little b, little a, little b. So that's where we get those two possible gametes. Now remember, Mendel's law of independent assortment says that these alleles, these chromosomes, can line up in any which way in this first step of meiosis. This would be metaphase of meiosis 1. So we can also get some other gametes. Now let me show you what I mean by lining up in a different way. I'm going to erase all this for now. We'll come back and make more gametes in a minute. And instead of putting the big A and the big B on the same side, I'm going to flip them. So now the blue is going to be over here, so the big B from the dad is going to be over here, and the little b from the dad is going to be on this side. Okay, so they can line up any which way, just as long as the B's are across from the B's and the A's are across from the A's, so that the same chromosome, remember we said one was homologous pair for chromosome 1, so those two chromosome 1s get together, and then the two chromosome 2s get together. And then again, when we split this, we look at this one more time, it's going to result in different gametes. Here's the first divide. Let's go through this real quick because this should be a review of what we already looked at. Okay, there's one side and the other. And then the second divide splits those sister chromatids apart for both of these cells. Draw the new ones in. Now we've got 4 possible gametes, and a blue in each, and a green in each, a yellow in each, and a pink in each. And remember, those colors represent those different alleles. So now I've got a little b with a big A, and I've got a little a with a big B. And there's our second set of possible gametes. So when I go back to the beginning and I say the linked genes that we have here, the unlinked genes that we have here could result in these 4 possible gametes, or these 4 possible egg or sperm cells. That's why. Okay, because they can line up in different ways in that metaphase 1 of meiosis through the law of independent assortment, and we could have all the different combinations of alleles that are possible, like sorting a deck, right? Like shuffling a deck of cards. Imagine these are all playing cards, and we shuffle them up together and put them in different groups of 2 randomly in different ways. Okay, those are all the possible combinations. Now that's supposed to be all review. So now we get into these linked genes. Now remember I said linked genes are found on the same chromosome, right? So it's like 2 alleles that are stuck together, or 2 genes that are stuck together. If we want to see what happens then, we need to diagram it a little differently. Oh, by the way, before we move on, notice again, there are 4 different gamete combinations. That's what I just said. 1, 2, 3, 4 different combinations of those letters. Okay, so here's our linked genes. As I said, you have to diagram it a little bit differently. And let's look at the original cell that starts before meiosis. This is the parent cell. In that cell, we've got 2 genes again, the A gene and the B gene, but this time they're located on the same chromosome. Okay, so maybe this is chromosome number 1. Chromosome number 1 has the gene for maybe eye color and height on the same chromosome, not on separate chromosomes. So A and B cannot be separated anymore. We can't shuffle them up independently into our deck. So if we look at how this goes through meiosis, things happen a little bit differently. So I'm going to start over with my original cell again. I'm going to give them different colors, just like I did before. Okay, so that we're going to line them up appropriately. For meiosis 1, remember they made sister chromatids already back in interphase. So now they're lined up from meiosis 1. Chromosome 1 across from chromosome 1, that's a homologous pair. The homologous pair separates in meiosis 1. Now we've got 2 cells. Okay, each of those cells has one of those chromosomes in it. Meiosis 2, the sister chromatids divide. So each of those becomes 2. And fill those in with the resulting chromosomes. And let's take a look at what alleles each one carries. The pink chromosome carried the big A and the big B. Big A, big B, big A, big B. The yellow chromosome carried the recessive alleles. All right? So the result of this particular time through meiosis was 2 different types of gametes, right? We could either have a gamete with completely dominant alleles in it, or a gamete with completely recessive alleles in it. Now, if we lined up these chromosomes in a different way in meiosis, so if we switched their spots, for example, we would still get these results of completely recessive gametes and completely dominant gametes. So it doesn't matter which way we rearrange these, because the A and the B gene are stuck on the same chromosome, we only have 2 possible gametes that are produced. Hopefully you can see that. Okay, we've got 2 possible gamete combinations. Oop, went too far, sorry. And the 2 possible gamete combinations, as I already showed you, are the completely dominant version, because that came from this linked chromosome, and the completely recessive version, that came from this linked chromosome. So when you have linked genes, there is less variety in the eggs or the sperm cells that are made, because those alleles can't be separated. They travel together through the gametes.